Welcome to This Explainer. Okay, I am genuinely so excited to get into this today because we are going to completely shatter the textbook illusion of DNA. You know the one, right? That perfectly still, symmetrical ladder just kind of floating in a void that we all had to memorize in high school biology. Well, the reality is way more dynamic. It's shape-shifting and it's hiding in plain sight across some incredibly different scales. We're talking about how this foundational molecule literally twists under pressure, how it hides out in unexpected cellular factories, and how it's actually letting us time travel through centuries of archived history. Let's get into it.
So here is our micro to macro map for today. First up, beyond the double helix. Then chloroplasts, genomes in green. And finally, mining museums for DNA.
Starting with Section 1, Beyond the Double Helix, Shape-Shifting DNA. We're going to begin at the absolute smallest scale, the atomic structure. And I want to throw a question at you right off the bat. Does DNA always look like a perfect double helix? We are so used to that classic Watson and Crick bDNA shape, but honestly, a perfect linear bDNA conformation is actually super unlikely to exist just like that inside a living cell. It simply can't afford to be that rigid. In reality, DNA actually has three major structural families. You've got aDNA, bDNA, and zDNA. Now, bDNA is your standard right-handed helix. aDNA is also right-handed, but it's wider, squatter, and usually shows up under dehydrating conditions. Plus, it literally has a hole straight down the middle of its axis. But then there's zDNA. This one is wild. It's a zigzagging, left-handed helix where the bases are actually flipped upside down compared to the backbone. The exact shape it takes depends entirely on the sequence of bases and its local environment. And the structural differences between these three families are really stark. For example, the base pairs per turn vary from 10 in bDNA to 11 in aDNA, all the way to 12 in that narrow zDNA. You might be wondering, why does DNA even need to be this flexible? Well, because there is nowhere near enough room for it to just stretch out and relax. In almost all cells, DNA has to be compacted by more than 1,000-fold just to fit inside. It has to flex, it has to twist, and it has to contort itself. To really grasp these physical quirks, we have to talk about something called the propeller twist. See, even when DNA forms those classic base pairs, the bases don't just sit perfectly flat against each other like rungs on a wooden ladder. Instead, they actually form an angle. Those paired bases twist away from each other, and it looks exactly like the blades of an airplane propeller. Now, while this twisting does create some mechanical strain on the delicate hydrogen bonds that hold the bases together, it happens for a completely brilliant chemical reason. It maximizes the hydrophobic or water-repelling interactions between the bases stacked on top of each other inside the helix. It's this beautiful, precise trade-off between bond strain and overall structural stability. Okay, I absolutely have to share this incredibly fun fact from the research. Get this. If you drop free adenine molecules into water, they will spontaneously stack themselves into single-stranded helices? It's amazing because it perfectly proves that the famous helical shape of DNA isn't some random accident. It is powerfully driven by those hydrophobic bases clustering together, actively trying to escape the water inside the cell. They literally twist to hide from the water. Moving on to Section 2, chloroplasts, genomes in green. Zooming out from the atomic level, we land at the cellular level to uncover a pretty surprising hiding spot for DNA. It isn't entirely locked away in the central nucleus like a vault. In plants and some algae, chloroplasts, the tiny structures that make plants green, actually have their very own specialized circular DNA. It's kind of like, well, think of the nucleus as the main public library, and the chloroplast is a specialized branch library. And this branch holds genetic code that is almost entirely dedicated to keeping the lights on. And let me tell you, the books in that branch library are absolutely vital. The chloroplast genome encodes some limited but unbelievably important machinery. The standout here is the production of an enzyme called Rubisco. Rubisco catalyzes the very first reaction during carbon fixation in the Calvin cycle. It is literally one of the most abundant proteins on the entire planet. It's the machinery that keeps our global ecosystems breathing and fed by turning atmospheric carbon into organic molecules. Talk about a heavy lifter.
But it doesn't stop at just photosynthesis, not even close. Just listen to this massive list of functions: carbon fixation, starch storage, synthesizing fall pigments, creating amino acids, making growth hormones, and even pumping out antioxidant defenses like vitamin C. These aren't just green blobs. They are incredibly complex, multifunctional energy factories, and they are guided entirely by their own hidden DNA.
Which brings us to Section 3, Mining Museums for DNA, The Rise of Museomics.
Now we're zooming way out across centuries to look at how DNA is being extracted from global archives. But first, we need to clear up a super common point of confusion. There is a very distinct difference between historical DNA, or HDNA, and ancient DNA, which is aDNA. Ancient DNA refers to those trace elements that are thousands to even 1,000,000 years old, naturally preserved in places like the permafrost. Historical DNA, on the other hand, comes from museum and herbarium specimens that were fortuitously collected by humans over the last 200 years. We're talking traditional voucher specimens just sitting on dusty shelves that were never, ever intended for DNA sequencing.
So, why does 200-year-old museum DNA even matter today? Because researchers are doing something completely mind-blowing with it. With recent tech advances and cost dropping, scientists are literally whole genome sequencing centuries-old dried bird skins, mammal pelts, and reptile mounts. And doing this allows them to build a massive spatiotemporal record. They can track historical genetic erosion, discover entirely new species that have just been sitting in a drawer for decades, and even trace the evolutionary origins of infectious diseases. It's unlocking modern biological mysteries using archival evidence. It's basically a genetic time machine.
But extracting this historical DNA is no walk in the park. For fluid-preserved specimens, scientists run into some pretty unique chemical roadblocks. You have to contrast traditional museum formalin-fixed specimens with modern clinical FFPE, that's formalin-fixed paraffin-embedded tissue. In a modern clinical setting, tissue is exposed to a really low 2 to 4% formalin concentration for a super rapid window, maybe minutes to 48 hours. But museum specimens collected back in the early 1900s, oh man, they were often exposed to 10% saturated formalin for days, sometimes even decades. That massive, prolonged exposure creates some incredibly unique DNA degradation challenges. It requires entirely different extraction protocols than what we use for modern medical samples.
And that brings us to the end of our explainer. But before we wrap up, I want to leave you with kind of a provocative thought. Think about those museum collectors back in the 1800s. They had absolutely no concept of the double helix. None. Yet the specimens they so casually preserved in jars and cabinets are yielding whole genomes right now. So if those 200-year-old specimens are revealing lost genetic secrets to us today, what will the DNA we are carefully archiving right now teach future generations centuries from now? The scale of genetic discovery really is limitless. Thank you so much for joining me on this journey today, and keep exploring.